for other people. Okay, so we have an advising assignment that is due Friday. If you have already been advised, this should be in your student kiosk. If you haven't been advised, you can send me an email and we can be flexible, but you need to show me that you have an advising appointment with somebody. Okay? So make an advising appointment. Either print it out or make an appointment by Friday. Because I mentioned this a couple weeks ago. Okay, and this is worth 5 points. So I have like 14 out of 42. So lots of people haven't given me that. We have 42 students in the class, and 14 people have given me their advising assignment. What?
There's only like 33 people here most days.
There's 42 people in the class. Some people... What? No. That has happened in my other class, but this... no. So, what? That's why I make you take that quiz at the beginning of the quarter to say that you have to go to you have to withdraw. Some people don't know that. They think that I magically withdraw them. Some people think that. Okay, so everyone saw this announcement about the advising assignment. So if you have been advised, you go to your student kiosk and degree audit and print out the paper and give it to me, or schedule an appointment with an advisor by Friday and send me an email about that. All right? So find your clickers. We're going to go ahead and start out with the clicker questions this morning. So you should have read through the end of chapter 33. I think the page numbers are off in the syllabus. 712. I noticed it this morning.
How are you?
I'm fabulous. Fabulous and amazing, as always. Come on in. We're getting started. Quick. It's 10 a.m. Well... Okay, so find your clickers. Oh, actually, I haven't started it. Darn it. I know. It's because I haven't started it. Okay, so we're going to close our books, and we're going to answer the first question quietly. So here you go. Here's your first question. Okay, it's time to answer the question. I'm going to close the question. Okay, so now you can chat with your neighbor. Did anybody leave this in the computer? This is for a wireless mouse. I'm trying. I think I'm giving up now. Okay, I'm going to reopen the question. Okay, it's time to answer the question. I'm going to close the question. All right, so we have a lot of choices on B, and then the majority of the choices on C. So let's talk about, D. So let's talk about choice B. So what do we know about nematodes? That pertains to what's here in the question. So is this true? Are they roundworms? Yes. Do they molt? Nematodes don't molt?
Yes.
Are they ecdysozoans? They have a cuticle. They molt. And they're pseudocoelomate. So the right choice is D. So porifera, what's wrong about this? They're not a coelomate. They don't have any tissues. Do they have a true gastrovascular cavity? No, they don't. They have an opening in the middle, but the individual collar cells take in the food. And remember, I said they then crawl around and give the food to the other cells. So even though they have an opening in the middle, what's that opening for? Well, it's for water flow, and it's not a digestive cavity. Okay? All right, so today we're talking about the deuterostomes, okay? So we're talking about the echinoderms and the hemichordates. These are two phyla. And then these three groups are all in phylum Chordata, the cephalochordates, the urochordates, and the vertebrates. So these are generally considered subphyla of phylum Chordata. So you're a chordate, and all those sea squirts that you saw on the field trip this weekend are in the same phylum that you are, okay? And this week in lab, some of you are seeing the cephalochordate. You'll see that tomorrow if you didn't see that yesterday. The little lancelet, the Amphioxus, is a cephalochordate, okay? So, these are the three phyla, echinoderms, hemichordates, and chordates. So find your carbonless paper and close up your book and tell me how you would define a deuterostome. So we've talked about this already. So especially if you had lab yesterday, you should be all over this. Okay, so what's a deuterostome? Okay, the name means mouth second. Okay, so what does that mean, Patrick? The anus forms from the what? Blastopore, all right? So what else do you know about a deuterostome? Deona? Yes, it's bilateral for the most part, okay? What else? But does that make it a deuterostome? Because protostomes are also mostly bilateral, right? So what's something else that makes it, specifically makes it a deuterostome? So talked about the mouth second, so the blastophore forms the anus. What else happens to make something a deuterostome? I'm asking Deona. Outpocketing of the gut. No, that's right. Okay, so the mesoderm forms, or you have outpocketing of the gut to form the coelom. Okay, what else for a deuterostome? Tia. The cleavage is indeterminate, so that means you can get what? Identical twins. Okay, what else? So those are the embryological traits. What else about the deuterostomes is unique relative to the protostomes? One more thing that we talked about that's not the embryology of it. Well, it happens during the formation, but no. Oh, you mean how the cells divide? Yes, radial cleavage. Okay, so we have radial cleavage, we have indeterminate cleavage, we have the outpocketing of the gut, the enterocoely. So those are the embryological characteristics. So there's one other characteristic that we talked about. Dorsal nervous system. Okay? So you guys should know that. Okay? You guys, that's just facts. That's recall. Remember when we're talking about types of knowledge, this is the simple stuff. Okay? So we have the dorsal nervous system plus these embryological traits that we've talked about. And here's the picture to remind you. So this figure is in Canvas. You want to find this figure. This might be one figure that you print out in color. I wouldn't print out the whole thing, the whole PowerPoint in color. So this is summarizing what we were talking about. All right, so today we're over here with the deuterostomes. We have radial and indeterminate cleavage. We have enterocoely, so the coelom forms via enterocoely, which is the outpocketing of the gut. And we have the anus developing from the blastopore. Okay? Any questions about that? All right. So we're going to start out by talking about the echinoderms, and you're going to see this in lab this week. You're going to dissect the sea star. You're going to look at some slides of the sea star. And you want to know what these names mean, because the name of the phylum helps you. So this means spiny skin. Okay? Echinodermata. Derm is skin. Echino is spiny. And they have an endoskeleton, all right? They also have, what's another unique characteristic about the echinoderms? That you're going to find one thing that tells you for sure, definitively, it's an echinoderm. What's another major characteristic of this phylum? The water vascular system. So that table that you're working on, page 48, you want to add a column about a unique characteristic. So these guys have a water vascular system. All right, so they have no segmentation. They're bilaterally symmetrical as larvae. They have radial symmetry as adults, usually in 5 or multiples of 5. So some books just call it radial symmetry. Some books call it pentamer symmetry. They have ossicles, so they have an endoskeleton made out. So they have calcium carbonate little ossicles. All right, some of them are in the skin, and some of them are under the skin. So in lab, you should have seen the ambulacral ridge, and it's like plates in each arm of the sea star. It's sort of like plates stacked in the dishwasher, the little ossicles. Okay, and they have this very unique water vascular system. Some of you watched the sea star feeding with its tube feed down at the dock on Saturday. When we passed around the little urchin in the baggie, you could see the spine sticking out, but you could also see the little clear tube feet. Okay? All right. So here's our picture of the sea star. The good news is when you dissect a sea star, you get 5 chances because each arm is the same. And the madreporite. So this is the opening to the water vascular system. It means mother pore. So it's got seawater, but it has other things in it as well. The mouth on these guys is underneath. Okay? And yes, you saw that at the dock. They have this ring canal, and then they have a canal in each of their arms. And each of these controls the tube foot. Okay, so here's a little tube foot. And if you look on this cross section up here, the part that's controlling it is called the ampulla. It's sort of like a little turkey baster, right? Squeezing water in to extend the tube foot, and if the tube foot's retracted, the ampulla would be full. Okay, it's Thanksgiving next week. Hopefully you all know what a turkey baster is. Someone in your house should. All right, so any questions about the morphology of the sea star? The arm is also called a ray. So in lab, it's labeled as the ray. It is more correct to call it a sea star. Starfish is commonly used. It's not a fish. It's an invertebrate. And so what do we see about the little sea star larvae over here? What kind of symmetry does this guy have? Bilateral. And the adult is radial. So if you have the 10th edition of the book, you have this nice photomicrograph of the Brachyolaria. There's actually multiple larval stages. In lab, you'll have a chance to look at a couple other ones. But this is the one that you have a picture of in your book. So they have these little ciliated arms that they use for feeding. And it's stained, okay? In nature, it's clear. All right? Anything that's, yeah. So here is another picture. This is from the 9th edition of the book. This is an urchin. And this larval stage is called a plutei. Okay? So, and this is a dead urchin. This is what's left behind. This is the test. Okay? So in the urchin, the ossicles are fused to form a test. In the sea stars, the ossicles are not fused. So when it's alive, you have the spines and the tube feet emerging from the test. Yes, Serena?
So when they die, do the spines naturally fall off?
They fall off. Yeah, so on the dock there was one that was dead that the insides have been eaten out by like a gull or something. So the spines are still there, but eventually the spines just kind of fall off. So you can find these on the beaches sometimes intact. Say that again? Yeah, it's not quite radial, it's kind of a flattened.
Like a roll.
Sure. All right. Okay, so here are some of the major classes of the echinoderms. Yes, Tom? Yes, a sand dollar is in the same class with the urchin. So this is, and we're not learning the class names, but they're called echinoids. And so a sand dollar is just essentially a flatter version of the urchin, and the spines are really short. And they usually live buried in the sediment partially. Yeah, so we have, these are called crinoids or sea lilies. These are the echinoids. So we have urchins, sand dollars, and something called a sea biscuit. These are the sea cucumbers. And these guys have reduced or absent ossicles. They don't have very many ossicles. Some species don't have any. And this is the sea star, and this is a brittle star. Okay? And there's actually a 6th class called the sea daisy, which is funky because it has 2 concentric canals in its water vascular system. But those are the major classes of the echinoderms. Yes, Kelsey.
So the cucumber has radial symmetry, like you cut it? Is that what's...
The cucumber has, so all of these things have radial symmetry. Yeah. Because it's still,
Like looking at it.
yeah, I mean, it does crawl. I guess if it had, it's usually considered radial. That one you could argue maybe it's bilateral. Depends on how you're cutting it, right? Other questions? All right. So the hemichordates. These are little creatures called acorn worms. They're close related to the chordates, hence their name. They're half chordates. So we're gonna, in a minute, we're gonna talk about the specific attributes of the phylum Chordata, and these guys just have the pharyngeal gill slits. They don't have the other features of the chordates, but they do have the pharyngeal gill slits. And these are little worms that are sessile suspension feeders. And there's a few different kinds. This is one that lives as a solitary organism. Okay. And then there are also colonial hemichordates, and this is what a colonial one might look like. So acorn worms, that's kind of the common name. There's not that many species of them. I don't remember exactly how many there are. And they are essentially named half chordates. Any questions about the hemichordates? It's a colony. See, it says tissue connecting colony members. So this one's colonial, and this is an example of an individual. So they can have either form. Okay? All right. So all right, so we talked about these two, the echinoderms and the hemichordates. And now we're going to talk about the chordates. All right, so the chordates. So you are phylum Chordata. Your subphylum Vertebrata. Your kingdom Animalia. All right, that's you. You should know how to classify yourself. Your class Mammalia. So we have 3 subphyla, and all of these organisms are bilaterally symmetrical with 4 shared features. So the book lists 3 because it's omitting the pharyngeal gill slits because that's shared with the hemichordates. But generally, when you characterize a chordate, you talk about these 4, and I don't know why they switched to 3. I'm going with 4. So we have pharyngeal gill slits because 3 are unique, I guess. The notochord. So this is what the phylum is named after, Chordata. The phylum is named after this notochord. This occurs in humans, it occurs embryologically, all right? But at birth, it no longer exists, okay? It's been replaced. It doesn't turn into them, but it's been replaced by those. So it's a stiff but flexible support rod. So this is the trait that's the most unique to the phylum. So if you find something with a notochord, you know it's a chordate. They have a dorsal hollow nerve cord. And in humans, your nerve cord is inside your vertebrae. It's protected. And a muscular postanal tail. Okay? So all of these are evolutionary innovations that are somehow evolutionarily advantageous to the organism. And so that's what you want to think about next in terms of your carbonless paper. So how is a notochord evolutionarily advantageous? Okay? So take a few minutes and write some thoughts about that on your carbonless paper. Okay, let's put that away and let's do this, okay? see if I want one green sandal and one red one. How you doing, Chris? Okay, so chat with your neighbor if you're sort of stuck and see what kind of ideas the person sitting next to you has. Okay, so let's hear from some different people. So what have you written down? What's the adaptive significance of the notochord? What's the notochord going to help with, John? Structure for protection, what? Of the nerve cord. So it's definitely giving it structure. But it doesn't necessarily protect the nerve cord, okay? So as we said, in humans, your notochord is resorbed during development, all right? So it's providing structure. What's it going to help the organism do? Do you think? Move, okay? Move or swim, all right? So it's going to help. With movement, especially for those that are swimming, okay? In organisms like us, we're not swimming as embryos, but it helps to, it has an embryological role in how it helps to form the structures, and we'll talk more about that when we talk about a little bit more cellular development that we'll do in the, I forget which quarter, second or third quarter, later in the year. All right, what about the muscular tail that extends past the anus? Stephen? Balance, okay. So it's going to help with swimming again, right? All right, what about the central nervous system with the dorsal hollow nerve cord? Jesse? Well, so vertebrates certainly have that, but so do the other two subphyla that are not true vertebrates. So this is advantageous for all chordata. So what's it helping with? What? Well, this is about the nerve cord. So this isn't about in humans we have a skull to protect the brain, but they don't have skulls, all of them. So what is this doing? Good. Okay, so essentially the nervous system is coordinating movement along the whole body. All right? Okay, so those are the three that are unique to the chordates. We said they also share the pharyngeal gill slits with the hemichordates. And if you looked at the, oh, did you have a question, John? Okay, we're going to look at some pictures in a minute. And you have pictures in your book, which you should have seen when you did the reading. But so if you did have lab yesterday and you saw the lancelet, what is the lancelet using that pharyngeal structure for? What? Eating. It's used for eating. It's used for feeding, primarily. Okay? So, the pharyngeal gill slits or the pharyngeal basket is used for feeding. There's gas exchange as well, but it captures food. All right? So you want to think about just not the sort of list of traits, but what their adaptive significance, their function is. All right? So, as we go through these chapters about the diversity of the phyla, you're learning the traits, you're learning the characteristics of the various phyla, but you want to think about how it all relates together and how it transpired across evolutionary time. And that's kind of the bigger, the picture, the higher order thinking skills. Yes, Jocelyn. Yeah. Yes. So we're going to look at some different examples. So it varies depending on the specific organism. Okay? All right. So any questions about this? All right. So here we see so we have our 3 different phyla, subphyla rather, and so this is, we have 2 of them here, right? We have the little sea squirt larvae. So we saw them stuck, right? We saw the adults, but they have this larval stage. And here you see the pharyngeal gill slits, which are used for filter feeding, and then the food moves to the gut, all right? And you see their muscular post-anal tail, and they have the notochord and the dorsal hollow nerve cord. So see here, it's not protecting it. See how the nerve cord's actually above that? So someone was talking about protection. So it's so all the attributes are here in the larval form of the urochordate. Okay, and this is our lancelet. So again, they're using this structure for filter feeding. Okay, so this one retains the structures throughout its whole life, and the tunicates or sea squirts do not. Okay, and we'll have a picture of that right here. So 2 common names, tunicates or sea squirts. Those are both common names for the urochordates. So the larval has all 4 of the features, and we just saw that in the picture we were just looking at, all of the 4 hallmark features of a chordate. But then as it metamorphoses, so it swims around as a little tadpole larvae, right? This guy looks a lot like a tadpole. And it's going to stick and metamorphose. So once it is attached, it changes. So they lose all of their chordate characteristics except, with the exception of that pharyngeal basket. And in some of the ones we saw down at the docks, you could see that. So the notochord places them securely within phylum Chordata. So a lot of you, when you first saw it, you thought it was a sponge. All right, and the way is to look for the 2 siphons of the adult. That's how you know it's a sea squirt. Because we weren't looking at the larva, we were looking at the adult. The sponges look like, the adults look like sponges or anemones. But if you see the 2 siphons, then you know it's a sea squirt. We also saw lots of colonial ones which were much smaller. A lot of them were orange, and you would need some microscopes to see the structures that would let you know that they were sea squirts or tunicates. All right, so here's a picture of an adult from the book, and you can see the 2 siphons, and it's the pharyngeal basket inside. So it uses that to catch its food, which then passes to the gut. And here, this is in your book. This is in an earlier chapter, and you can see the comparison of the little larvae. And this bright blue one is the one in the 9th edition. It's a different adult in the 10th. So if you go back to chapter 22, you can find this picture as well.
Are those in a different area?
What do you mean, are they in a different area?
Like all the ones down right off the sea?
So this is the difference. So at the dock, we saw a lot of the clear ones. Usually on those mucky buckets that weren't there, there's another species that's kind of brown and leathery. There's also lots of bright orange ones here that are also bigger. So we happen to see mostly the clear ones this particular time. This one's bright blue. It's probably tropical. Just like on magazines at the supermarket, they put the pretty supermodels. They put the most colorful things in the textbook. So people go ooh and ah. So I haven't seen blue ones here. I'm guessing it's tropical just because of how brightly colored it is. So I haven't seen blue ones around here. But it's a different species. Okay? All right, so here's your lancelet. This is the common name. In lab, the slide is called Amphioxus, which is one genus. It looks like a fish, but he's not a fish. They burrow in the sand and they suspension feed. Okay, so they use these structures to bring in food. And they are going to retain all of their chordate features throughout their life. Yes?
Are these closely related to lamprey? Just like
They look very similar to lamprey, but they're in a separate subphylum. Yep. So any questions about the cephalochordate? All right. So now we're going to talk about the our subphylum, Vertebrata. So here's your lamprey that you were just talking about. So we see that they have one feature, Eric. They have vertebrae that the little lancelet guy doesn't have. Okay? And we have jawless fishes, and then all of these organisms have jaws, and that's what this word means, and I'm not going to test you on that word, naphthostome. So we have the sharks and rays are chondrichthyans. We have ray-fin fishes. We have lobe-fin fishes, lungfishes. We have amphibians, and we have amniotes. Okay, and then there's another tree. So we're amniotes. All right, and you can see all these little red dots showing the various derived characteristics. So, for example, the ray-fin fishes have a swim bladder. And the swim bladder develops into the lungs eventually. All right? And so on. So any questions about the tree here? All right. So vertebrates have a skull. That's what's going to differentiate them from the cephalochordates and the urochordates. The skull protects the brain. That's its important function. And we now have the dorsal hollow nerve cord being protected by the spinal column, which is not the same as the notochord. Okay? The notochord actually goes away and a new structure, the spinal column, develops in the development of the organism. We have pharyngeal pouches present in all the embryos, and a lot of you had this on your comparative anatomy posters that we did a few weeks ago. We saw that. The structures were in all the fish, the amphibians, the reptiles. They all have these pharyngeal pouches. They become the gills in some organisms. They migrate into parts of your inner ear in humans. All right. So the notochord is present in the embryo, and its function is to help organize the somites. So you're segmented, and in lab you're going to see some segmentation. So segmented blocks of tissue are what we refer to as somites. The notochord itself disappears in the vertebrates. All right, so here's a fish. And it's showing you the anterior skull of the vertebrate contains the brain and other sensory organs. And then they're doing a cut right here, and they're showing you the cross-section. So here you see the dorsal nervous system, the vertebral column, and you have a large coelom. Okay? A large body cavity, and the organs are suspended in the body cavity. And that is going to be similar for all of the classes of the vertebrates. Questions? So we're not going to worry about the distinctions between the different lineages of fish. You want to know jawed and jawless. And then we're not going to worry about the chondrichthyes or in their own class. We're not going to worry about all that. We'll just call them fish. So we have sharks and rays have features that separate them. All right. So we're going to go a little bit, explain how these different groups evolved. So we've talked multiple times now about the Cambrian explosion. And we had all of the major phyla that are still alive on Earth today have representatives in the Cambrian fossils. So we had lots of different types of echinoderms. They look quite different than they do today. But we also had small fish-like vertebrates. So we know that at least by 530 million years ago, vertebrates, so this means million years ago, had a skull. They had skeletal elements to reinforce the gills, and they had a notochord of cartilage at that point in time. We have Hox genes, and we're going to talk about Hox genes and how they help direct development. Next quarter, we're going to learn in more detail, again, about the Hox genes. All right, so, and we said, why are Hox genes important? Shows common ancestry. So do we see these in other kingdoms? No. We talked about molecular traits that are unique to kingdom Animalia, and these Hox genes are one of them. We have genes with similar functions in plants, the body positioning function, but they don't have sequence homology with the Hox genes. Okay? So we're going to come back to this idea of a gene duplication event in a couple minutes. So we have these Hox genes providing positional information for the animal embryo. And so this is something that's new. So when I was an undergraduate, we talked about speciation, Hardy-Weinberg, changes in allele frequencies. That made sense to me. But I was like, how do you go from a jellyfish to an elephant? Those are really different organisms. And some people differentiate. Changes in allele frequency below the species level has historically been called microevolution. Some people have backed away from that term. How do you go from a sponge to an elephant? That's big, macroevolution. So when I was an undergraduate, the genetic toolkit that directs macroevolution was not yet understood. Okay? So we're going to introduce it this quarter. We're going to talk more about it next quarter. But there's this whole field of biology now that's called Evo-Devo. And it's the combination of evolution and development. All right? And a lot of it, not all of it, but a lot of it is related to these Hox genes, these homeobox genes. Okay? All right. All right, so let's answer this. Let's close our books and answer this silently, please. Okay, let's go ahead and answer the question. All right, I'm going to close the question. Okay, chat with your neighbor. Yeah. So we're trying to just hit the major phyla. So later, like when I was in school I think I was a senior, I took an invertebrate zoology class. So we learned lots more. in that table that I gave you, and what I present in the PowerPoint. Yes. So we're not doing the whole book. It's what's in that table and the PowerPoint and lab. All right, I'm going to reopen the question. Okay, I'm going to close the question. I think one more person needs to answer. All right. So the good news is you did a lot better the second time around. So the first time around we had 63% here. So B is the correct choice. So we were talking about the filter feeding is the pharyngeal basket, and then the gill slits. The tail, the nodal cord, the dorsal nerve cord help with swimming. Okay, so the nervous system needs to coordinate everything that's going on as the organism is swimming. All right? Okay. So, a little back to this question of how did we go from the jellyfish to the elephant. And these events have occurred that are called gene duplication events. And I think this is a situation where a picture is worth 1,000 words. So this figure is not in the textbook. All right? So I have borrowed this from a different textbook. So up here we have our hypothetical vertebrate ancestor with a single Hox gene cluster. All right? That just means that there's a part of the chromosome missing. All right? So this is the chromosome. So it's this is based on the current day, so they've sort of skipped something there. So but it just has one copy of those genes. Okay? So then there was a gene duplication event right here, first Hox gene duplication. So most invertebrates that are not the chordates have one cluster of Hox genes. Okay? Approximately 520 million years ago, a duplication event may have given rise to the vertebrates. Okay? So then we have the jawless fishes. So the hagfish and the lampreys, that are the jawless fishes, would have had two of these clusters of Hox genes. All right? And these new genes may have now direct the development of the backbone. Yes. And then we had a second Hox gene duplication event. And this is when we see the structures that we see with fish. Okay, so fish have jaws and limbs. They have fins, right? And the Hox genes that we have as vertebrates are homologous with the invertebrate genes. And in some of the other chapters in the book, you can see pictures of mouse compared with Drosophila. And we're going to come back to this topic again at the molecular level. But today we're just understanding it in a body structure level. All right? And so you can sort of think of this as when you have your original set of one or two sets of genes, you have, think of a factory. You have workers making X. And if you add on extra shifts, then some of the extra workers can specialize. So these genes some of these genes retain their original functions, and then the other genes are now free to mutate to take on new functions. And that's what's giving rise to your more structurally complex organism. Does that make sense to people? So that's, and the fossil record and the divergence of the sequences of the genes all support this hypothesis of two gene duplication events. So how did the duplication happen? So that's a really good question. So we talked about in plants how polyploidy is very common, right? Because some plants can self-fertilize, so the whole genome can become duplicated. And that happens very frequently in plants. And it obviously happened here. So some type of event occurred that allowed that would be similar to the polyploidy that we see in plants. Okay? All right, so it looks like we are out of time today. So we're going to finish this up on Friday and do the human evolution, the primates. So if you came in late, I've been talking for about 2 weeks now about your advising assignment, and it's going to be due by Friday. So if you've been advised, go to your kiosk and print it out. Otherwise, I want evidence that you have an appointment scheduled with an advisor, like a copy of the email. Go ahead and keep that. We're going to come back to that on Friday. Any other questions? I'll see you for lab tomorrow. See you later, guys.